Thank you very much. So normally I say thank you for a kind introduction, but in this case I just have to hope that it was a kind introduction. Thank you. So it's a great pleasure to be here. I thank you for the invitation to be here, and I'm going to talk through some of the work we've been doing over the last 4 or 5 years, where we've been doing magnetic resonance spectroscopy, and we have been developing methods to use MRS, magnetic resonance spectroscopy, to measure Gaba, and we've been trying to see what that can tell us about inhibitory function within the brain. The other one. So my background is in the physics behind magnetic resonance, and at this stage within my lab we have sort of 3 main areas of interest. We still do a lot of work in technical areas, trying to improve the methods, make them more robust to various different kinds of things. And then we try and apply the methods that we develop to understand how the healthy brain works and how the unhealthy brain goes wrong. What happens when things are not working as they should. And so between these 3 areas we're going to be sort of moving around this picture through the talk today. So I know that people here have very different backgrounds, maybe to each other and to me. So I sort of have a few introductory slides that I've added in, and hopefully they'll be of interest. So it seems to me that there are 2 ways that we can use MR, in particular in vivo magnetic resonance, to do science, if you like. So the main way that imaging is used is to take pictures of, in particular my interest is in the brain, and from those pictures to be able to interpret signals and signs and biomarkers, basically changes that we can associate with certain features. And so this is sort of what I've called signal signs and biomarkers, the idea that we can just image the brain in some non-specific way and we might see evidence of something happening. Whereas another way that we can use MR is to really target specific processes, target specific chemicals within the brain, and use MR as a technique to help us understand what is happening in the brain, how those things are happening, why they're happening. And this sort of more focused way of using MR is how I think of magnetic resonance spectroscopy. We are able not just to image the water signal, which is within the brain tissue, but to detect small signals from metabolites that are present within tissue, and by individually detecting metabolites, we can probe individual processes within the brain. So in this sort of preliminary section, I want to talk a little bit about neurons, about synapses and receptors. From my background in sort of physics and chemistry, these are very much things that I'm learning, so I'm aware that I may be talking to people who know far more about this than I do, so if my explanations are rather simplistic, forgive me, but I think certainly they will give you a sense of where I'm coming at this from. And then I'm going to introduce what Gaba is doing within the brain, and in particular its role in tuning, and then to start to motivate some of the studies that we're doing relating Gaba to behavior. So what is Gaba? So Gaba is the principal inhibitory neurotransmitter in cortex, and I sort of really highlight this statement as being incredibly important. This is the whole motivation for what we're doing, and for some fraction of you this will be entirely sufficient motivation for why we would want to look at Gaba, and so I guess those people can sort of have a sleep for the next 4 slides or so, and the next 4 slides are going to be unpacking this statement, talking about what we mean by an inhibitory neurotransmitter, and therefore motivating why we would like to look at Gaba. I myself have stood up Oh my goodness, the slides are going to be a disaster. So never move your slides onto someone else's computer and hope they'll work. So I've stood up at an international conference and said that GABA is the main excitatory neurotransmitter in cortex, and this was maybe down to a lack of preparation, but far more than that, it was down to a complete lack of understanding as to what a neurotransmitter was, what excitation was as opposed to inhibition, and no real understanding of the neuroscience that we were trying to probe with the physics. So GABA is not the main excitatory neurotransmitter. GABA is inhibitory. So what do we mean by that? So you're probably familiar that there are cells within the brain that are called neurons, and in essence you can think of a neuron as an information processing module. And so the neuron has inputs which come in on the dendrites, and then you can think of the cell itself as making some kind of decision based on those inputs whether to put out an output, and that output goes down the axon. And so if we think about inputs and outputs and the cell as a decision-making machine, once the cell has made a decision, then the output will be transmitted at a synapse to another cell, and within the synapse the output signal from one cell appears, and there is then, if we're talking about neurotransmitter synapses, certainly there is a neurotransmitter released from the presynaptic cell, from the first cell, which then docks on the second cell and then starts to become a part of the input signal to that other cell. And so this first cell has made a decision to put an output signal out, and this second cell then will start to make a decision based on its inputs whether to put an output out itself. And if we zoom in again to the synapse, within the synapse there are receptors, and so when a neurotransmitter when neurotransmitter is released from the presynaptic cell, it will then be released into the synaptic cleft, the area between the two cells. It will dock on a receptor on the postsynaptic bank of the synapse. And then for most of the neurotransmitters that I'm going to be talking about today, these receptors work as ion channels. So when the neurotransmitter docks, the ion channel becomes open, and there can be a flow of charged particles into the cell. And in the case of GABA, it is negative ions that are flowing into the cell. And so here immediately with this idea of negative ions flowing, we have introduced the idea of charge, and it is charge, electronic charge, which is the currency in which the cell is making those decisions. So there is a voltage across the membrane, and based on the total sum of the different charge flow events that happen at the inputs, the cell will then make a voltage-based decision on whether to put out an output. And so here you can start to see how we have two opposing processes, excitation and inhibition. Excitatory events will tend to make the cell more likely to give an output, and inhibitory events will make the cell less likely to give an output. And the main excitatory neurotransmitter within cortex is glutamate, and the main inhibitory neurotransmitter is GABA. And these two are opposite in effect because GABA allows negative ions to flow into the cell, and glutamate allows positive ions to flow into the cell. And so in terms of this voltage decision that the cell is making, they are opposite in effect. And so the majority of cortical neurons are glutamatergic, they are excitatory. That is, when they have an output signal, they will release glutamate. But a substantial fraction, 15 to 20% of the cells, are GABAergic inhibitory neurons. And so I apologize, and I will apologize once and then try to stop apologizing. These slides are a little bit funny. So GABAergic inhibition has a key role in controlling and shaping activity within cortex. So what do we mean by this? Well, let's imagine for a moment within the touch system. So if there is a point stimulus on the skin, then this point stimulus on the skin will be represented by a peak of activity within the cortex representing that area of the skin in primary somatosensory cortex. And so here in this cartoon, we have this cartoon of a peak of activity, and this will be excitatory activity associated with that input. But the way that cortex is wired up is such that that peak of activity also causes a suppression of signal adjacently within cortex. So there is this so-called surround suppression phenomenon whereby whenever you have a peak of activity, adjacent cortex will be suppressed. And it turns out that this surround suppression phenomenon happens widely within cortex, and it is reliant on GABAergic inhibition. So if we compare here situations where we have inhibition from where we don't, and now we are talking about 2 points of contact very close together on the skin. So these 2 points of contact will each be represented by a peak of activity within cortex. And if we have this lateral inhibition, this surround suppression, which is mediated by GABAergic inhibition, then the points of activity associated with those two inputs will be separated, whereas when we take away the GABAergic lateral inhibition, the points of activity are far less well separated. They are almost blurred together completely. And so when we think about what the person is perceiving, when there are 2 points of contact that are separately represented within cortex, this will be perceived as 2 things touching the skin, whereas if the GABAergic tone is reduced and they basically blur into 1 peak, the person will not be able to tell that there are 2 points of contact. They will just feel it as 1 point of contact. And so in this case, we might say that GABAergic lateral inhibition improves spatial tuning. It improves how accurately the cortex can represent the position of contact within the skin. And why does spatial tuning within cortex matter? Well, spatial tuning within cortex is going to be the thing that determines how well you can interpret the signals that are coming into your body. So here again, in different form, here is the idea that we have a peak of activity and lateral inhibition leads to suppression of surrounding cortex. And if we have less GABA, we don't see the wells of negative activity adjacently within cortex, but also the central peak of activity is broader. So this lateral inhibition within cortex helps to sharpen the peak of activity, and that sharpening of the peak of activity improves representation of the position of the stimulus in cortex. And this kind of wiring is not specific to somatosensory cortex, and it's not really specific even to position within somatosensory cortex. In general, there will be GABAergic connections which help to tune cortical responses to stimulus properties. So the more within any particular parameter dimension, the more lateral inhibition we have, we might expect the representation of that property to be improved. And that's really the motivation for these studies, and in a sense the explanation as well. So I want to talk at the beginning about how we measure GABA with MRS. Hopefully by now you've been convinced that that might be an interesting thing to do. I'm going to talk about some studies that we did where we looked at individual differences between healthy people and individual differences, particularly in this GABA signal, and to what extent these can be related to individual differences in brain activity and individual differences in functional performance. And I'm going to start to talk about some of the current work that we have going on where we are starting to apply some of these approaches in neurodevelopmental disorders. And so I'm going to talk about some developmental disorders, but this is, I think, a generalizable approach that can be applied in any clinical domain, really. So you are probably all familiar with MRI as an imaging methodology, and MRI uses the signal from water, and there's a lot of water in the brain. It uses the signal from water and encodes spatial information into that signal so that we can image the water, and it then relies on the water in different kinds of tissue behaving differently to encode contrast within the images. So, for example, we can have contrast based on the MR relaxation properties of water, or we can have contrast based on the diffusion, the physical motion of the water. We can encode a lot of different properties into that water signal. Now, if we throw away that very large water signal, then we can see things from other compounds within the brain. So it's not just water we're looking at, we are looking at hydrogens within the water, and we're looking at the nucleus of the hydrogens within the water. And it turns out that a whole load of other interesting molecules, in particular small organic molecules within the brain, have hydrogens in them, and we can detect the hydrogen in those molecules just as we can with the water. It's just that these molecules are much lower in concentration, whereas some high fraction of the weight of the brain is water. These metabolites that are doing the biochemical reactions within the brain are present at tens of thousands of times less concentrated than the water. But we can still detect them. So we throw away the water signal and we say, what else can we see? So if we say, take a small piece of brain tissue, don't take it out of the brain, leave it in the brain, but let's imagine, if we say, what signals can I detect from this small region of brain tissue, we will get a spectrum that looks something like this. And here we have various peaks that I've associated with different metabolites. So here, for example, there is a strong signal associated with N-acetyl-aspartate, and various other peaks which are associated with other metabolites. And there is this separation along the x-axis because of something called chemical shift. Basically, different chemical things have slightly different frequencies of signals, and so we get different peaks for different chemicals. Now, immediately, this shows some of the promise of MRS. We can separate different chemical species out into a spectrum, but it also shows one of the very great limitations of MRS. These different chemicals are separated along this axis, but they're not separated very well. If you look at the fundamental width of these different signals, it is actually quite wide when you compare it to the total amount of axis that we separate all the signals along. And when you think that I have, I don't know, if I'm generous, I might say there are 15 peaks there that I can really say are peaks, and I can associate them with a number of different metabolites. We all know that there are far more than 15 different small molecules within the brain. So what we have here, if you like, is a problem whereby there is too much information in this spectrum that isn't separated well enough. And if we ask the specific question of how do we measure GABA within the brain, well, you can see I'm being a little bit optimistic. There may be some small hump just down here that might be associated with GABA, but really GABA cannot be well quantified with this kind of approach. These signals are not well enough separated to quantify all the things that we would like to. And if we ask, where are those GABA signals? There's one down here at 1.9 ppm, and it's maybe contributing a little bit to this shoulder. And there's one here that optimistically might be in that gap. There's one here at 3 ppm that's definitely underneath a much stronger signal from creatine. And so if we want to measure GABA, we are going to have to find a way to separate it from all the other stuff in the spectrum. And so this separation procedure that we talk about is called editing the spectrum. We are going to take that spectrum that has a lot of information that's very poorly separated, and we're going to throw out a lot of the information so that we have much less information in the spectrum, but it is much better separated. And this is the editing procedure. So basically we take advantage of the things we know about GABA. So we know that there is a GABA signal over here. We saw that in the previous slide. And we know there's another GABA signal over here. And it turns out that the physics is kind to us, and there is an exchange of information between these two GABA signals. And so we can use that phenomenon to separate the GABA signal at 3 ppm, which is very small, and underneath this large peak from creatine, we can separate that GABA peak from the creatine, because the GABA peak under here is talking to a GABA signal over here, and the creatine peak here is not. So if I do an experiment where I apply a frequency selective pulse to just this bit of the spectrum, which I can do, then it is going to have an impact on the shape of the GABA signal that I see here, and it's going to have no impact on the shape of the creatine signal that I do here. And so I can do a subtraction experiment, where I do one experiment where I just get all of these signals, and I do another experiment where I get all of the signals again, but this time I tickle the GABA system. And when I subtract those two things, I'm going to see what was the effect of that editing pulse. I'm going to get an edited spectrum that only contains those signals that are affected by the editing pulse. And when we look at what this looks like in vivo, it looks something like this. So immediately, rather than there being a large number of peaks, you can see we've reduced the information content down to a very small number of peaks. And you can think of these in two terms. So these peaks are all of those peaks that are affected by the editing pulse. And here, at 2 ppm, there is a strong negative signal from NAA. And this is sort of trivial. The NAA signal is affected by those editing pulses because this is where we're putting the editing pulses. But then far less trivially, there are remote effect peaks over here, whereby this GABA signal at 3 ppm was affected by the editing pulse that's put over here, because there is this transfer of information within the GABA molecule. The GABA peak at 3 ppm is coupled to the GABA signal at 1.9 ppm, and so remains in this different spectrum. So this was some data I acquired during my postdoc in the sort of very first work that we did looking at GABA. Other people previously had looked at GABA, and the methods that I'm presenting are not methods that I have designed. These are other people's methods that we have adopted and tried to improve. And this data was acquired from a 3 by 3 by 3 centimeter cubed voxel in about 15 minutes. And still we're working within that regime. Our typical acquisition parameters are 3 by 3 centimeter cubed voxels acquired for about 10 minutes. So that's how we measure GABA. What can we do with it? So I'm going to talk about combining GABA MRS with functional imaging. Hopefully you're all sort of somewhat aware of functional imaging. We can ask a person in the scanner to do some particular cognitive task, and if we design the experiment correctly, we can start to pull out spatial representations of which brain regions are engaged in that task. This is maybe a very simplistic way of looking at functional imaging. And we can do functional imaging with fMRI, with functional magnetic resonance imaging. And we can also use sort of direct detection techniques like MEG or EEG. So MEG directly detects magnetic fields associated with neurons firing in the cortex. EEG, which is very closely related, detects the electric fields associated with neurons firing in cortex. And MEG and fMRI are very closely related in terms of what we're trying to do. We're trying to look at which bits of the brain are doing what. But they're very complementary in the terms of their strengths. fMRI is better at localization, really getting down to where things are happening, but is very poor at temporal resolution. It's very bad at saying when things are happening, whereas MEG is very good at saying when things are happening and not so good at saying where. So these things are comparable. And in sort of in psychology departments across the world, these methods are being used, and they are telling us, I think, a lot of interesting things. But it's surprising, I think, on some level that there has been such rapid uptake of these methods, and when you consider that we don't fully understand the generative mechanisms of the signals that we're studying to infer things about brain activity. And in particular, one thing we know really very, very little about is what are the particular drivers of individual variability in these signals. So we did a series of experiments where we used a very simple low-level visual task to start to probe individual variability in some of these signals. So I will zip through the acquisition parameters because I think that nobody is particularly interesting. These are standard Gaba measurement parameters. For the functional imaging, we had people fixate on the red dot, and then occasionally within the lower left visual field there would be this striped grating would appear. And during on periods the striped grating would appear, and during off periods the grey background would appear. So there's no overall difference in the amount of light that's coming into the eyes, it is just the spatial pattern of that light that is changing. And it turns out that if you set the spatial frequency of this grating at 3 cycles per degree of visual field, it will very efficiently stimulate a high frequency oscillation within primary visual cortex that we can detect with MEG. And so by high frequency I mean in the 40 to 70 Hertz band, what we call gamma band oscillations. And we acquired MEG data for about 20 minutes and fMRI data with the same visual paradigm for about 8 minutes. Now, the functional imaging data from the MEG, we were able to reconstruct spatially to show that there was activity happening in, for a left visual field presentation in right occipital lobe in primary visual cortex. And when we had this activity in primary visual cortex, we could not just spatially locate it, but we could also look at the temporal features of that response. And so, time-locked to the onset of the stimulus, there was so I should explain these plots to start off with. So the y-axis here is frequency. May or may not be legible. The y-axis is frequency, so down here at the bottom are low frequency things, and as we go up, these are high frequency things that are happening. The x-axis is time, so this time frequency plot lets us look at which different frequency band features are happening at what point during the activity. And so we are applying our stimulus for 2 seconds. At the beginning of the stimulus, there is a broadband response. So there is things happening at a lot of different frequencies. But then later on during the stimulus, there is a very narrow band response at a finely defined frequency that continues for the duration of presentation of the stimulus. And so this narrow band response, we can see these 2 people are 2 different people, and immediately we see something interesting happening. It seems like the response in this person is happening at a lower frequency, about 45 Hertz. Response in this person is happening at about 60 Hertz. And we can integrate that signal for the duration of the presentation, and we can now present the signal change, or the thing that was coloured here is now the y-axis, and the x-axis here is the frequency, or what was the y-axis up there. And we can see, we can confirm that yes, one person, the grey person, had a response that was down at 40 Hertz, and another person had a response that was up at 60 Hertz. So we have a feature of response to a very low-level visual stimulus that is different between different people. And it's not just different between different people, it is different in a way that is stable over weeks. So the 2 lines for the black subject were acquired 5 or 6 weeks apart, and for the grey lines again 5 or 6 weeks apart. So this is a feature of the brain's response to a tightly controlled visual stimulus that's different between different people and is different in a way that's stable between people. And over a cohort, we had gamma responses at a frequency ranging from 40 to almost 70 hertz. And this was where we started. So the MEG people in the lab in Cardiff had discovered this feature, and they started looking at, well, what might that be caused by? What is going on? What is causing these gamma band oscillations? And when you look in the literature, there is modeling literature which suggests that these high frequency gamma oscillations are made by local, by local I mean within cortex, networks of excitatory and inhibitory cells, and it may be that the frequency of these oscillations is set by the balance of excitation and inhibition. So we set about testing the hypothesis that differences in GABA frequency, differences in the amount of inhibition, might explain these individual differences in the frequency of this response. And so we measured GABA in occipital lobe as well. And when we combined these data, we were able to see a correlation between the concentration of GABA and the frequency of these gamma band oscillations. And the correlation was in the direction where those people with higher GABA concentration tended to have a higher frequency oscillation. We also saw a relationship in these data between GABA concentration and bold signal change. And this time it was in the sense that those people with higher GABA concentration were the same people who had a smaller bold response. And the tightest correlation statistically of all between these three features was between the two functional imaging metrics. The bold signal correlated with the MEG gamma frequency. Now, we have a lot of things which correlate with each other. It turns out that not everything is correlating with everything, so we are not seeing correlations between GABA and the amplitude of the gamma response. We're not seeing correlations between gamma and the amount of grey matter within the voxel or within the occipital lobe, or with the thickness of cortex in these different people. So the correlation between GABA and gamma is very provocative. We do not fully know the neuronal origin, or what is the sort of cellular substrate of these gamma oscillations, and the direction of the correlation that we see is in line with some modelling work that's been done by Brunel and Wang. The relationship between GABA and bold is, I think, very surprising. If ahead of time you said, what are the main features of the functional response that differ between different people, then I think most people would say it is something to do with the hemodynamic properties of tissue. So the bold response is a surrogate marker of activity. So we have a change in neuronal activity which causes a change in blood flow and blood volume, and then that change in blood flow and blood volume changes the relative concentrations of oxy and deoxyhemoglobin, and because these things are magnetic, they then impact the behavior of the MRI signals. So we have this sort of multiple steps from the neuronal activity, which is what we're really interested in, to the thing we're looking at, the fMRI signal. And I think most people might say that any relationship, any individual differences in the bold signal might be related to this stuff in the middle, the properties of the vasculature, the properties of the blood flow and blood volume. But it does seem to be that individual differences in bold are reporting on something to do with individual differences in neuronal properties, because the concentration of the inhibitory neurotransmitter GABA has to be a neuronal property. So we got very excited about these data, and we tried to send it to a bunch of journals. And along the way, one of the editor comments that came up is he said this may be a neuroimaging epiphenomenon, which I think was a phrase he was very pleased with himself for saying. But I think what he meant by that was, if this thing is happening in your scanners and it stays in your scanners, I don't really care. I will only care about this thing if you can show me that it gets out of your scanners and has an impact on the world, has an impact on behavior, that it can relate to some individual difference in behavioral performance between different people. And so we sort of said thank you and sent it to a different journal. But we then got to thinking, and that set the tone for the next phase of the work. So we got to reading, and you don't do much reading when you're looking at visual performance and you are talking about the inhibitory neurotransmitter GABA before you come on the orientation discrimination literature. And really at the early days when they figured out that GABA was an inhibitory neurotransmitter, they were right back then doing orientation experiments. And there's a long literature of direct cellular recordings in cat visual cortex and in various animals. And it turns out that within primary visual cortex in mammals, there are individual neurons that are tuned for orientation. So if you give a visual stimulus that has this orientation, that neuron might fire, but if you give it with this orientation, that neuron will not fire. And it turns out that that tuning for orientation of those cells is dependent on GABA. So not just is the location of stimuli within the visual field encoded by position within cortex, there is also other features encoded. Orientation is one of them, and the tuning for that parameter is dependent on GABAergic inhibition. And so we set about trying to say, well, how well can people perform an orientation discrimination task? And discrimination is a sort of a typical psychology way of getting at how well does cortex represent this property. So you show two slightly different things and say, what is the smallest difference between these two things that this person can notice? So we present one striped, I realize I'm favoring the right-hand side of the room, but I can't point over there. We show one striped grating, and then after a short delay we show another striped grating with a slightly different angle. And the person then has to say, was the second grating clockwise or anti-clockwise relative to the first? And if the task is easy, they will get it right. And if the task is very, very difficult, they will get it right half of the time. And somewhere between those two extremes there is the threshold of performance whereby they're getting this right about 75% of the time. And it's that threshold, that point at which people go from being able to do this to not being able to do this, that we want to get at. And there is this idea within psychophysics of stepwise tracking, whereby we start off with the task being very easy, and people get that right. And as they get right answers, we start to make the task harder and harder and harder, until at some point they stop being able to do it. And so you get some wrong answers. And at the point where you hit wrong answers, you make the task easier. And then you start getting some right answers and you make it harder. And the task itself starts to oscillate about the threshold of performance where you're getting it right approximately 75% of the time. And so you can then average the turning points at the end and interpret that as the person's ability at this task. And when we combined this with measurements of GABA in primary visual cortex, we saw a relationship, just focusing on the left-hand picture here, we saw a relationship between concentration of GABA and the oblique orientation threshold in degrees. And those people with higher concentration of GABA tended to be the same people who could do better at this task. They had a lower threshold. And so this really nicely lines up with the hypothesis at the beginning that tuning for orientation within cortex is associated with GABAergic inhibition. Now, I've focused very much on this side because this has a significant result. I haven't talked about the other side. There is an interesting thing about orientation discrimination that is well known, is that everybody is very good at this task about the vertical. And you can explain this both evolutionarily, that trees are vertical and monkeys need to know what a tree looks like. You can explain this experientially, that when I am walking around every day, I want the verticals to remain vertical. And if they're not vertical, then I need to adjust my balance, else I fall over. So there is something special about the verticals, and to a lesser extent there's something special about horizontals, but it's really the verticals where everyone does really excellently. And so it may be that there is no relationship between discrimination at the vertical and GABA concentration, or it may be that there is a weak relationship that's sort of drowned out by the noise within the data. We don't know. But certainly this observation is significant even after we control for the fact that we have done two separate observations of behavior. So this, I mean, this now is remarkable, I think. So we have asked a person to do something, and we have measured how well they do it, and we have taken them on a separate day and put them in the MRI scanner and made a measurement of one of millions of brain chemicals, and there is a correlative relationship whereby that measurement of one chemical in the brain is correlated with the behavior. And when you think of all the different steps that are in between those two things on the macro scale and the molecular scale, I think this is a really remarkable result. So we, at this point, I suppose, had a paradigm that we could go and run with. I was within a psychology department, and people in psychology departments know how to measure behavior. And we now have a potential marker within Cortex of how well the Cortex can do things. And so we started to try and translate this approach to different modalities. At this point, I'm going to ask what my time limit is. So I normally talk for an hour, but I've been going slowly because somebody said that I could overrun an hour. Would you prefer the hour version or the longer than an hour version? I don't know. Okay, well, the longer version. Okay, so I won't, because the shorter version was going to be the long version but faster. So that's probably preferable. Okay, so we then said, okay, can we do this in a different domain? Is this telling us something special about the visual system, or is this something that's generalizable across cortex? And so we started looking in the tactile domain, the touch domain, and to cut a long story short, there's a lot of evidence that GABAergic inhibition, as we saw in these earlier slides at the beginning, GABAergic inhibition is related to the representation of stimulus properties in somatosensory cortex. So sorry, I had a brain blank. Touch is represented in somatosensory cortex, and for a number of reasons, we looked at a frequency discrimination task. So how well can people tell the difference between an oscillation at one frequency and an oscillation at a different frequency? Excuse me. And again, we did this in a tracked, stepwise kind of task, whereby the task itself homed in on the threshold of performance, the point at which somebody kind of could do the task and kind of couldn't. And the way we did it, one of the stimuli was always 25 hertz. Another stimulus was either higher in frequency or lower in frequency. And we did tracked experiments on a sort of a lower and an upper arm. And coming from top down, people got to within about 6 hertz of the correct answer before they started getting it wrong. And from the bottom up, they got to within 5 or 6 hertz again. And so the actual numbers are not interesting. What is interesting is that there is some degree of variance between people. And so the question is, can we relate GABA concentration measured in primary somatosensory cortex to performance at this task? And it will hopefully be unsurprising, since I'm talking about it, that the answer is yes. And again, just like we saw in the visual system, we have a correlative relationship whereby those people who had a higher GABA measurement in this brain region were the same people who performed better at the task. They had a lower discrimination threshold. And so the answer, at least partially to this, does this generalize, is maybe, I suppose. Is this something special about visual cortex? No, it's something that we can also show in somatosensory cortex. And actually, I have to say at this point, there is a bit of a disclaimer. So these GABA measurement regions are large, and they are much larger than the cortex that is doing the task that we're measuring. And so in particular, in this case, the GABA measurement we're making is from a mixture of primary somatosensory cortex, which is on the posterior bank of the central sulcus, and primary motor cortex, which is on the anterior bank. So we call this a sensory motor measurement. So even despite the fact that there is a lot of cortex that isn't doing somatosensory things in that voxel, we still see a correlation. Interestingly, we also did measurements in the visual cortex for these same subjects, and it turns out that this somatosensory behavior, this tactile behavior, does not correlate with the GABA measurement in the occipital cortex. So it is not true that there are some people who are high GABA globally within the brain and other people who are low GABA globally within the brain. It seems that this individual difference in the GABA concentration is local to some extent, in that the individual differences, the GABA concentration in somatosensory cortex does not correlate with the GABA concentration occipitally. So at about this time I moved from Cardiff in the psychology department to Johns Hopkins in School of Medicine, and we had some results that we were very excited about, and we said, how can we apply this paradigm? And really I think this is a powerful thing. We have a brain marker of inhibitory neurotransmission, the GABA-MRS measure, and we have ability to measure behaviors that we know are related to inhibitory processes within cortex. And it turns out that there are a very large number of disorders where inhibitory processes are thought to be disturbed to some extent. And I'm going to present some data from a series of neurodevelopmental cohorts, but there are also evidence that GABA is disturbed in various psychiatric disorders, various neurological disorders, in aging and in neurodegenerative disorders. So really this approach, although I'm showing you it in children, I believe generalizes. And what I think we can do is, by combining measurements of behavior with measurements of the brain chemicals, I think we can make this more powerful than just measuring a brain marker or just measuring a behavioral marker. We really have some explanatory power, and the combination of these metrics hopefully can be more powerful than them individually. So we have developed a battery of tactile measurements, and the data I showed you previously was a frequency discrimination task, which is just one of many different things you can do with tactile psychophysics. So we have developed a battery where we're now doing about 10 tasks. So we have a reaction time task and a choice reaction time task. These are very easy. We have detection threshold tasks that ask the question, what is the weakest thing you can detect? And inhibitory processes within cortex are related to signal to noise within cortex, so detection is interesting. And then we have some discrimination tasks, both amplitude discrimination, which one is stronger, and frequency discrimination, which one is vibrating at the faster frequency. And many of these tasks we are doing because we are interested in inhibitory processes. So the reaction time tasks aren't really specifically related to inhibitory processing, but they're an important indicator that people are attending and that they have listened to the instructions and understood the instructions. So first of all, I'm going to show you some data in healthy adults and typically developing children, and then I'm going to start to compare the typically developing children to the children with developmental disorders. And so here on the left, in the reaction time plot, you can see that the simple reaction time in the white circles is faster than the choice reaction time. There is a cost associated with that choice in terms of how fast you can do things. You have to both respond and make a decision. And that is true both for the adults and the children, that choice reaction time is slower than simple reaction time. For the detection threshold, we have two different ways of measuring detection threshold. So one is to apply a stimulus at a constant amplitude and ask the question, can you detect that stimulus? Or actually, we apply it to one of two fingers and the person has to say, well, was it on that finger or that finger? and thereby we can track that experiment, we can lower the amplitude of that until it becomes almost not detectable. Another way of measuring detection threshold is using a dynamic detection threshold task. Here we are basically going to have a very low amplitude stimulus going on one of these two fingers that slowly increases in amplitude until it becomes detectable, and then the person says, ah yes, I feel that stimulus, that's on that finger. And so these two different ways of measuring detection threshold are complementary, but they are specifically different in terms of how they are going to impact cortex. And so if you think about this dynamic detection threshold task where we have a stimulus that is increasing in amplitude throughout its duration, let's imagine that the static detection threshold is here. Okay? So early on in the experiment, we have a vibration that is happening, and it's happening below threshold. And it turns out that subthreshold stimuli are going to more efficiently stimulate they are going to more efficiently stimulate inhibitory circuits than excitatory circuits. And so by preferentially stimulating excitation, by preferentially stimulating inhibitory circuits within cortex, we are then going to increase the threshold. Basically, if we make things, if we add additional inhibitory input, then the threshold is going to be placed up. And so then when we do this dynamic task, the threshold that we measure will be raised. And that threshold is raised because that early subthreshold stimulation stimulates inhibitory circuits more. And here again, in healthy adults and in typically developing children, we see that they perform significantly worse at this task than this task. And this comparison between the two tasks is something that we can interpret as a marker of inhibitory function. So this is maybe an example of how we can use comparisons between tasks to specifically probe inhibitory processes. I'm going to jump across these tasks. But again, we're doing some amplitude discrimination tasks, and there are 3 different amplitude discrimination tasks that we do, and the performance at the different tasks is different in a way that reflects inhibitory processes. So why are we interested in Gaba in neurodevelopmental disorders? So in autism or autism spectrum disorders, there are mouse models that are GABA related. There are gene association studies that show GABA genes are affected in autism. And there is also in ASD a very high incidence of epileptic seizures. And this seizures, or if you like, excessive and uncontrolled activity within cortex may be related to a failure of inhibition. In ADHD, attention deficit hyperactivity disorder, this is essentially defined as a deficit in behavioral inhibition, in the ability to control motor behavior. Now, it's not obvious that behavioral inhibition is mappable onto cortical inhibition, but we do believe, at least to some degree, that that is the case. And then there are brain stimulation measures like short intercortical inhibition, or SICI, which are reduced in ADHD. So this is a TMS-based measure of inhibition that has been shown to be reduced in ADHD. And Tourette's, again, there's a wide range of evidence that GABA is impacted in Tourette's syndrome. So these are 3 distinct but overlapping neurodevelopmental disorders. There are high levels of comorbidity between these 3, and in all of them there is evidence that the GABA system may be involved to some extent. I'm not saying GABA is the only problem, but there is, I think, a GABA component to these. And I think to some extent a lot of the unsolved developmental and psychiatric disorders are going to be multifactorial, they're going to be multi-system, and the reason why we haven't solved them yet is related to their complexity and the number of factors that are in play. Within autism, this is something that sort of has seeped into popular understanding with television shows and such. Autism is defined as a deficit in communication and social development, but it's also very widely seen that there are sensory impairments, and up to 95% of parents will report that their children with autism respond differently to sensory inputs, and repetitive behaviours may be related. One example that our neurologist collaborator gives of a typical feature of autism is this tag in the shirt idea. So in my collar there is a tag, there is a label, it's there. Every time I move it is stimulating somatosensory receptors in my skin, but I am not perceiving it the whole time. There is some mechanism within my brain that has habituated to this constant input and which considers it to no longer be of interest, no longer to be useful to give it any of my attention. And the ability to habituate, if you like, to these kind of constant inputs is something that is frequently impaired in autism. A child with autism will not be able to ignore that label. It will continue to bother them until it is cut out. There are other sort of common features of autism associated with sensory and in particular somatosensory impairments. And one of the things that we're really selling in terms of why we think that doing good, rigorous psychophysics is a good way of getting at these processes is a lot of the clinical literature at this point looking at sensory features of autism has been done through questionnaires, and they're normally teacher or parent report questionnaires. So you are not just you have the problem that you are talking about one person's interpretation of another person's unexplained behaviour. You also have a problem that they do not seem to be discriminating between sensory responses and emotional responses to sensory events. So there is something happening within primary somatosensory cortex which may or may not be different, and then there are emotional processes that lead out of that also may or may not be different. And we think that by doing psychophysics we're really able to probe whether those early sensory responses are indeed changed, or whether it's just all frontal, whether it's just all about the emotional response control. Just as a brief introduction to Tourette's syndrome, it is a syndrome which is characterized by motor and vocal tics, so either repetitive and uncontrollable movements or vocal outbursts, if you like. And these tics, these repeated behaviors, have associated with them a premonitory urge. So they are not unforeseen, they are just uncontrollable. So a person will know that a tic is coming, they will not be able to suppress that tic, and in a sense they will find relief from being able to tic and get that premonitory urge to be satisfied. And again in Tourette's, even though this is a motor disorder primarily, children with Tourette's do exhibit different tactile impairments, and they again show different difficulty habituating to sensory input. In ADHD, we have been able to show, and I'm just sort of going through an overview of some results now, we've been able to show that Gaba is reduced in attention deficit hyperactivity disorder, so this is with MRS, and this was just an MRS study. We have been able to show in Tourette's, this is data, I beg your pardon, we've been able to show in autism, this is unpublished data as of yet, that Gaba seems to be reduced in sensory motor cortex, but not in occipital cortex, and these are our data, but other people have shown the same thing. And we've been able to show in Tourette's syndrome that Gaba is reduced. That children with Tourette's syndrome have reduced Gaba, again in sensory motor cortex. So here you sort of see some of the power of what we can do with the MRS, but also some of the limitations. So we have, if we just do an MRS measure of GABA, then we can say, well, yes, it's gone down in these disorders. But we don't get any texture to that. We don't get any real explanatory power as to what cortical processes are affected. Inhibition is doing a ton of different things within cortical circuits. There's feed-forward inhibition, there's surround inhibition, there are lateral things, there's all sorts of inhibitory processes. And so what we hope is that by combining this MRS, which yes, does differentiate between groups, by combining that with behavioral measures, we can actually get some context to which particular inhibitory processes are affected. Okay, I'm going to skip through this bit. So, looking at one particular behavioral thing, this detection threshold that I talked about, we saw earlier that in typically developing children, the dynamic detection threshold was worse than the static detection threshold, and we interpreted this difference as a marker of inhibition. When we look at this in autism spectrum disorder, there is no difference. People do not do significantly different at one task than the other. Effectively, there is no difference associated with this feed-forward inhibition in the task. And again, in Tourette's syndrome, we see that there is no difference between performance at this task. In that dynamic task where you have early, low amplitude stimulation that is below threshold and which is preferentially stimulating the inhibitory circuits, that's not having an impact in ASD and Tourette's in the same way as it does in typically developing children. So this is nice. This is a behavioral marker of impaired inhibition in autism spectrum disorder and Tourette's. But it's also limited in the fact that there isn't really anything different between what's going on in autism and Tourette's. So is there a behavioral marker that we can see that starts to discriminate between the different disorders? And here we're going to look at amplitude discrimination, and in particular amplitude discrimination as a simple task, and amplitude discrimination when preceded by an adaptation stimulus. And in typically developing children, this adapting stimulus makes people worse at the task, as you can see there. And this worsening of behavior, again, we are interpreting as a marker of inhibitory processes, but a different inhibitory set of processes. And now, in autism, you again see that there is not a significant difference between performance at those two tasks, which is consistent with impaired inhibition. Again, in Tourette's, there is no difference between those tasks, which we can interpret as impaired inhibition. But what we see now is that in Tourette's, in the baseline condition, people are performing the same as typically developing children. They just don't get worse at the task when you apply the adapting stimulus. Like typically developing children get worse, children with Tourette's do not. Whereas now the children with autism spectrum disorder seem to be much worse to start with and do not get further worse when you apply the adapting stimulus. So now we think we have a behavioral marker that is starting to discriminate between these different groups. So I'm going to sort of wrap things up at this point and take any questions. This work in developmental disorders is ongoing, and we don't have all of the answers, but we certainly think it is interesting. So hopefully I've been able to convince you that we can measure GABA concentration with MRS, and that this seems to be able to measure functionally relevant differences in GABA concentration. In particular, it can explain inter-individual differences in functional neuroimaging signals and in sensory discrimination tasks. And we can detect reductions in GABA concentration with MRS in various neurodevelopmental disorders. Tactile psychophysics is a quantitative methodology that allows us to measure behavior and might allow us, through combinations of tasks, to probe specific inhibitory mechanisms in somatosensory cortex, and we have been able to show that alterations seen in ASD, autism spectrum disorder, and Tourette's syndrome. And really, I think that the promise of this work that we're trying to move towards is that MRS on its own can tell us whether Gaba is reduced or not. But it's not giving us any functional information on which aspects of GABAergic inhibition are affected. It's not really giving us context, it's not giving us texture, it's not giving us the detail that we really need to know if we're going to understand how does that relate to what's actually going on in these disorders. And so what we hope is that by combining it with quantitative measures of behavior, we can start to get at separable inhibitory processes within cortex. So I want to finish by acknowledging a lot of things. First and foremost, I want to acknowledge my collaborators. This is work that sort of far exceeds my personal expertise, and so we rely heavily on clinical collaborators. I rely heavily on the people in my group who have done most of this work, my boss Peter Barker for supporting me, various people in Cardiff who helped to establish the early work in healthy people that prefigured all of this that I'm talking about now, and obviously the funding that we have currently to carry on developing these methods, disseminating them to groups like you here, and to carry on try and see what we can do with these methods and what they can tell us. And thank you all for your attention.